I'm going to introduce Shelly Flagel. Shelly, as you could see from her title, there's professor in our psychiatry department and also has an appointment in the Michigan Neuroscience Institute, of which she was recently the acting co-director, actually. Um, Shelly actually trained here. She was a graduate student and a postdoc here and has worked on motivation reward, particularly in the context of addictive behaviors and learning behavior. But I think what has really distinguished her work has been her focus on sort of individualization of rather than treating everything as a uniform construct. Why do some individuals, including apparently inbred strains of rats, actually respond differently under these different conditions? And she's done a lot of interesting work in this respect. So, Shelley, thanks very much for giving a talk. So,
Yeah, thanks so much for having me. This feels odd. My office is under construction and I haven't given a Zoom presentation from home in a long time, so hopefully there won't be too many more technical difficulties, but thank you all for being here nonetheless. I'm excited to tell you a little bit about our recent work and some future directions, hopefully if we have time. Okay. So you look at the screen and I'm sure there's certain cues or symbols on here that may capture your attention more so than others. It's late afternoon, you may be wanting that afternoon cappuccino, or you may be looking forward to that after dinner beer this evening, or dessert. And all of these, the ability of these cues to capture your attention have to do with your prior experiences at these establishments. And so we know that cues can attain the ability to control behavior via classical Pavlovian associative learning processes. The value attributed to a reward cue determines the extent to which it can control behavior. So we know that some cues are attributed with predictive value, simply signaling the availability of reward, whereas others are attributed with incentive value and can instigate aberrant motivational states and lead to maladaptive behaviors. And Roger already told you that I'm interested in individual variability in how one responds to cues in the environment. And we've actually known that there's individual variability in associative learning since Pavlov's times. So he actually described different personality traits amongst his dogs that were his research subjects, and noted great variation in how they responded to rewards and reward-associated cues. And so he described two variations. The first, he said, was with exaggeration of the excitatory and weakness of the inhibitory process, and the second with the predominance of the inhibitory and weakness of the excitatory process. And what I hope to tell you today is that I think that we can actually capture those processes using a rat model of individual differences in response to reward cues. And so the way we do this is we simply put rats into a conditioning chamber where they're exposed to an illuminated lever for 8 seconds. That lever retracts, and then they receive a food pellet in an adjacent food cup. So importantly, this is classical Pavlovian conditioning. The rats don't have to do anything for the reward to be delivered. Yet what we see happens is these very distinct condition responses. So on one extreme, we have the sign tracking condition response, or the phenotype sign trackers. When they see that cue, when it's presented, they go after it, they approach it, they grasp it, they chew on it, they gnaw on it as if it were the reward itself. And then when it retracts, they go to the food pellet and get the food reward. And the other extreme here are goal trackers. Goal trackers see the cue for what it is. It's a predictor, and they go to the food cup and wait for food to be delivered upon cue presentation. And so we can quantify these behaviors, and the way that we do this is we look at the vigor of the response, the amount of contacts with the lever, the probability or the likelihood of approaching the lever, and the latency. How quickly do they go to that lever cue when it's presented? And then on the other hand, for goal trackers or goal tracking behavior, we can look at the vigor with which they enter the food cup or the magazine when that lever is presented, the probability to approach, and the latency. And what's important to note here is that if you look at the learning curves, that is a change in behavior over time, it's quite similar between sign trackers, which are shown here in blue, and goal trackers with their goal-directed behavior shown in orange in the bottom. So that is, they're both learning a condition response at the same rate. It's not differences in learning. It's not differences in the sense that one may be smarter than the other within this context. It's just simply the topography of the condition response that differs. And that topography tells us that for both sign and goal trackers, the reward or the cue is a predictive stimulus. That is, it elicits a conditioned response for both. But for sign trackers, we also know that it's an incentive stimulus. It's imbued with incentive motivational value, and that's what makes it attractive and approachable. And then on the right here, you can see that we measure these behaviors and characterize the phenotypes with the Pavlovian Condition Approach Index. So this is a composite index score taking into account all those metrics I told you on the previous slide, and where we can look at values that are above 0.5 to 1 and characterize individuals as sign trackers, and those less than negative 0.5 to negative 1 we characterize as goal trackers. And typically we characterize them across 5 sessions, each session consisting of 25 trials of cue reward pairings. So why is this important? It's because this is one of the first animal models that allowed us to dissociate predictive versus incentive learning processes and thereby be able to parse the underlying neural mechanisms. Interestingly though, as I've noted before, if you caught it, we know that a cue attributed with incentive value can also trigger aberrant motivational states and lead to maladaptive behavior. And so what's fascinating to me is that we can simply characterize rats based on these 5 sessions of Pavlovian conditioning as sign or goal trackers. And we know then that we're capturing this neurobehavioral endophenotype of relevance to a number of neuropsychiatric conditions. So for example, we know that sign trackers are more impulsive on tests of impulsive action. They have attentional deficits akin to ADHD. They show enhanced cue-induced fear response similar to PTSD, and enhanced cue-induced relapse behavior to drug seeking, and dysfunctional checking behavior similar to obsessive compulsive disorder. On the other extreme, we know that goal trackers are better able to use contextual cues to regulate their response, so they show enhanced context-induced drug-seeking behavior and enhanced context-induced fear responses. What I'm going to start this talk with in terms of data is demonstrating how we've been able to utilize this model to parse the neural mechanisms that contribute to predictive versus incentive learning processes. And so one of the first questions that we wanted to ask in this regard and to utilize this model for was to ask, what is the role of dopamine in predictive versus incentive learning processes? So as you may know, the role of dopamine in reward processing has been quite controversial for a number of years, and that controversy is not going away, but I'll tell you a little bit about what we've learned with the sign and goal tracker model in that regard. So the first study I did teamed up with Paul Phillips and Jeremy Clark at the University of Washington, where they helped me use fast-scan cyclic voltammetry to look at dopamine response in the nucleus accumbens during these cue reward learning. And then I'll follow up with some more recent data from Amanda Iglesias, who is a recent graduate student in my lab, who's now doing a postdoc at UC Davis, where we used optogenetics to follow up on those initial studies. So the scientific premise here is based on work from Wolfram Schultz and colleagues back in the mid to late 1990s. And what Schultz and colleagues showed was that, and this work was initially done in non-human primates and monkeys. And what they showed was that if you look at dopamine activity, you can see that initially dopamine is going to enhance in response to a reward. But if you then show a cue that predicts that reward, the dopamine response shifts from the reward to the cue itself, in this case, the CS, a conditioned stimulus. And so we're then in a good position to model this similar setup using our same paradigm and to ask whether or not dopamine encodes the predictive or the incentive value of reward cues. So if Schultz was right and it encodes a predictive value of reward cues, then dopamine in response to the lever cue should be similar between sign and goal trackers. I'm going to show you some videos to illustrate what we did and what we saw. And so this is a sign tracker. This is a dopamine trace in the nucleus accumbens on the bottom from fast-scan cyclic voltammetry. The lever comes out and you see this big increase in dopamine in this sign tracking rat when he's interacting with the lever. Lever retracts, goes and gets the reward, and you don't see much, excuse me, of a response in dopamine once they consume the reward. On the other extreme here, you have a goal tracker. And the lever comes out, orients to it, and then goes to the reward. And you can see when he looks at the lever, not much is happening. And it's not until he consumes the reward that you see the big increase in dopamine in the goal tracking rat. So we were able to quantify this, and these are voltammograms showing you that that shift in dopamine from the reward to the cue is happening in sign tracking rats and not in goal tracking rats. What that then told us then, as I told you, if it was attributing to the predictive value, we would have seen that shift in both sign and goal trackers. We only saw this shift from the reward to the cue in sign trackers, indicating that the dopamine response is actually encoding the incentive value. To follow up on that, we did a number of pharmacological studies, as did Ben Saunders and Terry Robinson's lab. And this is using a strain of rats that we know, these red high responder rats that we know are also sign trackers. So it's rats with an inherent propensity to sign track. We give them systemic flupentixol blocking dopamine receptor, activity of dopamine receptors, and you can see that we're blocking the sign tracking response. Interestingly, we do the same thing to goal trackers, and you see that we're blocking the goal tracking response. But what was especially interesting and telling was that when we removed flupentixol, they were no longer under the influence of the drug. You can still see that that sign tracking response is blocked, whereas the goal tracking response recovers. So what this showed us then was that there was evidence of having learned the goal tracking response, where attributing the incentive value to the cue and learning the sign tracking response was completely blocked under the influence of dopamine receptor antagonism. Like I said, Ben Saunders followed up on these studies doing local antagonism in the nucleus accumbens core and showed similar effects where you're blocking the expression of a sign tracking response and not a goal tracking response. So then more recently, we followed up on these studies using optogenetics. And so if dopamine encodes the incentive value of reward cues, then presumably optogenetic inhibition of dopamine precisely at the time of cue presentation should block the development of sign tracking behavior. And so to do this, we used tyrosine hydroxylase Cree rats provided by Carl Deiseroth and then the colony maintained by Huda Akil here at Michigan. And what was interesting about these, and what really actually opened up this opportunity for us, was that we found when characterizing these rats that the entire population in our hands was skewed towards sign trackers. So this then allowed us to ask the question, what happens if we inhibit dopamine at the time of cue presentation? Are we attenuating that propensity to sign track? And so to do this, we did surgery for implantation and viral expression of the optogenetic receptors. And then what we did is we specifically inhibited dopamine activity during cue presentation during the first 3 sessions of Pavlovian conditioning, and then we removed that laser inhibition and the laser was off for the following training sessions. Again, this is work done by Amanda Iglesias, and what we saw here, this is showing you the first 3 sessions when the laser is on, when that optogenetic inhibition of dopamine neurons is happening during cue presentation. And here what you can see is that we're completely blocking that tendency to sign track when we inhibit dopamine in the experimental animals relative to controls. If you look at food cup directed or goal tracking behaviors, we're not seeing a difference between those that receive laser inhibition and those that didn't, the control animals. What you can see here, this is session 3 at the end of the 3 sessions of laser inhibition, just to show you the individual variability. And again, you can see that we're essentially squashing that variability in the experimental animals that received laser paired inhibition during cue presentation. And then here, once you remove that laser inhibition, now there's no longer inhibition occurring. What's interesting is that now these animals that previously received laser inhibition show that they're perfectly capable of learning that sign tracking response. Now you're releasing that inhibition, and they presumably are attributing incentive value to the cue. Again, you don't see much of a difference in food cup or goal tracking directed behaviors, although you do seem to have lifted the brakes a little bit on their activity at the food cup as well. And then this is just showing by session 6, after 3 sessions of not having that laser inhibition on board, there's essentially no difference between the experimental groups and the control groups. So we were lucky to team up with Alvin Chu and Christian Burgess and do kind of a deeper dive into the behavioral analysis using deep lab cut. So for those that don't know, deep lab cut allows us to essentially train and identify very specific behaviors occurring during these sessions. And we were specifically interested in activity of the animal and activity directed specifically to the lever or the food cup that we're not able to assess with the boxes themselves. So not just looking at contact with the lever or head entries into the food cup, but rather general locomotor activity and whether
who's ever not muted, please mute yourself.
Okay, got it. And whether or not they're approaching the area of the lever or the food cup. And so here is an example of tracking these animals. This is a control animal. Inhibition is occurring, but it's still interacting with the lever. And then this is an experimental animal where you can see the lever is out and they really show very little interest in the lever, and they go to the food cup and wait for the food to be delivered. And so like I said, we were able to, there we go. We were able to get a really nice picture of what was going on with the behavior using deep lab cut. And so here on the left, you can see general activity maps showing that here you can see the control animals are their general patterns of activity, especially during lever presentation, is quite different than the halorhodopsin animals
Mhm.
that received inhibition. So here you can see, hopefully you can see my cursor, they're essentially spending more time at the food cup than they are at the lever.
So
What I really, yeah, yeah.
shall me just interrupt briefly? So does this sort of imply that actually goal tracking is sort of the default behavior?
Um, Not necessarily. I don't. Why would you think that? Oh, I think you're muted, Roger.
You said these are usually sign tracking animals, but you've sort of blocked the
Oh,
sign. Yeah, yeah,
right.
right. You've sort of blocked the sign tracking by blocking the dopaminergic stimulation. So, and they're going to the reward instead. So does that seem more like seems like you've turned sign trackers into goal trackers to some extent?
Yeah, I mean, if you look at, if you remember, let's see if I can go back. That didn't, I mean, so here during inhibition, you can see that they're really not different. But I think I'm hesitant to draw many conclusions about goal tracking itself, especially the role of dopamine in goal tracking, given that these animals had that predisposition to sign track. And so initially they are pre-trained in the sense that, yes, they all may show this initial tendency to go to the food cup because they're pre-trained just for food delivery. But I'm hesitant to say that that's the default because we don't see that in other, that doesn't necessarily, it's not supported by other neurobiological mechanisms and other animal strains that we use. I think here we can say a lot about sign tracking. I'm not sure we can say much about goal tracking given that these animals tend not to goal track. However, based on this here, and again, hopefully you can see my cursor, it does look like those that are not spending time at the lever due to laser-induced inhibition or laser-paired inhibition are spending more time at the food cup.
Yeah, I think Cindy has a question.
Yeah.
Yeah, I mean, so my question was actually sort of in the opposite direction, which was that, when I take a look at the control animals, it's actually a little surprising to me because they seem way more obsessed with the lever, which, they would have to learn then with the actual incentive. Just, when I'm taking a look at the approach behaviors, just, taking a look at how high the purple dots go up the y-axis for lever versus magazine. For the controls, it looks higher to me for the lever, which in some ways is a little surprising. But when we did our smokers, we actually found that they obsessed with their lever too. So
Yeah, so these animals, they're a transgenic strain, the TH rats, that we happen to show and know that have a tendency to sign track. So that's why you're seeing that in the control animals, they have that tendency. And then with the laser paired inhibition, you're kind of blocking that tendency. So that's what you're seeing with this individual variability on the far right. Yeah, so then, so these are the heat map that like I said, are telling and not anything we've been able to look at previously. And then we look at very fine-tuned resolution at head orientation to know if they're orienting more towards that lever cue when it's presented versus the food cup. And we're seeing differences there also during lever presentation. And then finally, as Cindy noted, the approach routes towards the lever versus the magazine. And what's, I think, most important, one of the more important things with this analysis is that it showed us that any differences in activity were very specific to that lever cue presentation period. So by inhibiting dopamine at the time of cue presentation, we weren't having necessarily longer term effects on general locomotor activity. It was very specific to that cue presentation period. So to summarize what I've told you so far, we've used this sign and goal tracker animal model to demonstrate that a shift in dopamine from the reward to the cue actually encodes and send a motivational value of the reward cue, not the predictive value, as Schultz had put forth. We also show that inhibition of dopamine neurons in the VTA, specifically during reward cue presentation, attenuates the development of sign tracking behavior. And all of this leading us to the conclusion that dopamine appears to be critical for the attribution of incentive value to reward cues and the development of sign tracking behavior. So back to Pavlov, we're going to keep going back to his wisdom throughout to kind of keep us oriented. But essentially, what it seems to be is that dopamine may be playing a critical role in that exaggeration of the excitatory responses. And so the next question was, what beyond, of course, it's not just dopamine, although sometimes I like to think it is. We wanted to ask the broader question of what neural circuits are encoding predictive versus incentive learning processes. And so this was a cohort in my lab of graduate students and postdocs who played a really big role in these studies over the years, and also talk about some work in collaboration with Martin Sarter and Young-Soo Kim in the Department of Psychology here in Michigan. So this is a complex subway map that we've created based on our work and that of others who have looked at what's going on in the brain of sign trackers versus goal trackers. And in general, what has led us to believe is that goal trackers have this enhanced top-down executive function or inhibitory control, whereas sign trackers have this exaggerated bottom-up subcortical motivational processes. And I already told you that dopamine seems to be a critical player in that regard. But in terms of the circuit level, what's the paraventricular nucleus of the thalamus has emerged as a key player. And so the PVT is very nicely located to integrate information from subcortical areas and cortical areas and send it to the nucleus accumbens to guide motivated behavior. It initially popped out in our Q-induced CFOS studies repeatedly, showing that sign trackers have essentially twice that number of activation in response to the cue relative to goal trackers and unpaired control animals. And so after multiple studies of it popping out on the old school films, I couldn't ignore the thalamus anymore. And then when I did an additional analysis, it highlighted the importance in terms of regulating sign and goal tracking sign trackers and goal trackers. And so essentially what I did is I looked at correlated cue-induced neuronal activity within phenotypes. And so here you can see in sign trackers what I call my correlation wheel, and you don't need to pay attention to the color or the thickness of the line necessarily, but those regions that are connected have correlated cue-induced neuronal activity. And here in sign trackers, what you can see is that almost all of the neuronal activity that's correlated is subcortical. Whereas in goal trackers, you can see engagement of the cortex, and specifically in both, what you can see is a PVT, or the paraventricular thalamus, is playing a role. So in sign trackers, that PVT shows correlated activity with the nucleus accumbens, and specifically the shell, whereas in goal trackers, you see this correlated activity with cortical regions like the prelimbic cortex. And so this led us to hypothesize that specifically neurons from the prelimbic area to the PVT may play a role in encoding predictive value and perhaps inhibiting incentive motivational processes, whereas the bottom-up encoding of incentive value may be coming from areas like the lateral hypothalamus and nucleus accumbens as well. And so specifically, I'm gonna quickly summarize a number of studies that we did and then walk you through a few of them. So we know that when we looked more specifically in terms of neurons that are activated in response to the reward cue in sign trackers versus goal trackers that are communicating with the PVT, we found that while they both engage these corticothalamic striatal circuits, sign trackers seem to engage hypothalamic thalamic striatal circuits to a greater extent than goal trackers. So that bottom-up encoding is occurring to a greater extent in sign trackers than goal trackers. We know that you can lesion the PVT and enhance sign tracking and attenuate goal tracking behavior. And we know that lesions of the lateral hypothalamus or orexin receptor antagonism within the PVT both seem to attenuate the propensity to sign track. So what I'm going to walk you through now is the role of what we found was the role of the PRL to PVT pathway using a chemogenetic approach. And so one of the first questions we asked regarding the circuit is what happens if you turn on this top-down cortical control? That is, what happens if you stimulate these neurons from the PRL to the PVT? So to do this, we initially characterized animals as sign or goal trackers. Then we did surgery to insert the dread virus specifically in those neurons. And then we rescreened them for Pavlovian condition approach behavior, and then administered clozapine N oxide to activate those dread receptors or vehicle. And so this is just showing you that we use a dual vector approach for selective expression of dreads in the neurons that project from the prelimbic cortex to the PVT. And we get selective expression. And when we do so, what you can see here in sign trackers, this is the rescreening and then the test. And unfortunately, there was a difference during the rescreening, but that difference during the test is more pronounced. So meaning that when we administer CNO during the test session, when we're turning on that top-down inhibitory control in sign trackers, we're attenuating the tendency to sign track. In goal trackers, when you do the same manipulation, we don't see an effect. In contrast, next we asked what happens when you turn off that top-down PRL to PVT pathway. So now we're using the inhibitory drug receptors. When you administer clozapine oxide, CNO, you're inhibiting or turning off that top-down control pathway. You do that in sign trackers and you see no effect. You do that in goal trackers and now all of a sudden they start to attribute incentive value to the reward cue and start to show a tendency to sign track. So next we wanted to ask, I don't know if you caught it before on the slide that I showed, but we know that there is output from the paraventricular nucleus of the thalamus to the striatum. I noted that before. And we know that it can actually regulate dopamine release independent of the VTA. So now we wanted to ask, if we're manipulating this PRL to PVT pathway, are we doing anything to dopamine in the nucleus accumbens, especially knowing that we're affecting the tendency to sign track and that sign tracking behavior is dependent on dopamine. So here, whoops, maybe. Here you go. So what you can see is when we turn on that top-down corticothalamic pathway, you can see a tendency for a decrease in dopamine in the nucleus accumbens. So again, we're not specifically manipulating dopamine, we're manipulating that PRL to PVT pathway, but showing that we're affecting accumbens dopamine. And the effect was even more pronounced in goal trackers. When you inhibit that top-down PRL to PVT pathway, you're actually enhancing dopamine in the nucleus accumbens. We looked at a number of other neurochemicals, again in collaboration with Martin Sauter and Young Soo Kim, and dopamine was the only one that showed differences. And what was interesting is that when you're inhibiting that pathway and increasing dopamine in the goal trackers, you seem to be tightening up that correlation between the Pavlovian condition approach index, that tendency to sign track, and dopamine in nucleus accumbens. So what I've told you so far in terms summary about this, the PVT, is that the PVT appears to be a critical node in the regulation of cue-motivated behaviors. We showed that top-down input from the prelimbic cortex appears to inhibit incentive learning, and output from the PVT to the accumbens seems to be critical in this regard, as we're showing a change in dopamine as well. And it's likely that output from the PVT to the accumbens that may be the common integrative pathway that's mediating cue reward learning in this regard. So back to Pavlov, I told you that I think dopamine may be playing a role in these excitatory processes. I think the PRL to PVT, this corticothalamic circuit or pathway, may be playing a role in the inhibitory processes. But again, we know it's not that simple. So where else are we looking? Another pathway that we're looking that may play a role in these inhibitory processes is that from the prelimbic cortex to the nucleus accumbens. And so why this pathway? Well, we know that this pathway plays a role in inhibitory control. This has been done for decades. A number of groups have shown that this pathway specifically is involved in inhibitory control and in impulsive behaviors. We also know that dopamine in the nucleus accumbens is critical for sign tracking behavior. I told you that, and also for these processes, essentially like inhibitory control, that tap into impulsive behavior. And finally, we know that neuronal input from the prelimbic cortex to the nucleus accumbens can directly regulate dopamine activity. So a very quick summary of what we know about the PRL to nucleus accumbens pathway to hopefully set the stage for why we're targeting this particular pathway. So we really wanted to ask, what is a neuromolecular mechanism within this pathway that may be playing a role in the tendency to sign track or in the attribution of incentive value to reward cues? And so this is work that's been led by Princess Felix in the lab. And specifically, she's a graduate student in the neuroscience program. And specifically what she wanted to ask was, does a glucocorticoid receptor within the PRL to nucleus accumbens pathway play an important role in incentive learning? And so why focus on glucocorticoid receptors? Well, one of the first studies that I did, having come from a stress-related research background, was to look to see if corticosterone, the main hormone mediating the stress response, differs between sign trackers, goal trackers, and another phenotype, the intermediate phenotype, essentially. And what we showed was that corticosterone increases following a Pavlovian conditioning session in all animals, but to a much greater extent in sign trackers than the other behavioral phenotypes or the control animals. This wasn't that surprising. Leading up to this work in the 80s and 90s, we knew that corticosterone plays a role in reward processing as it does in stress responsivity. We then later showed that you can administer cort and you can increase the tendency to sign track, or you can block glucocorticoid receptors and attenuate the tendency to sign track. And this is systemic administration of cort or glucocorticoid receptor antagonists. And again, we knew from older studies that CORT can act directly on the nucleus accumbens dopamine. So this is systemic administration, again, corticosteroid self-administration in animals, showing that you can affect accumbens dopamine release. And you can administer a GR antagonist and reduce endogenous dopamine. So there's this interaction between glucocorticoids and dopamine, and we know that CORT in particular and GRs play a role in sign tracking. So then to ask the question, does the glucocorticoid receptor within this pathway play a role in incentive learning? We teamed up with Jim Herman at the University of Cincinnati, where he was generous enough to give us his transgenic Sprague Dawley rats, that we call the GR glucocorticoid receptor CRISPR rats, that allow us to knock down GR in a region-specific or pathway-specific manner. So the way that we did this is we again used a dual vector virus approach, where we're putting a flipovirus into the nucleus accumbens core that's going to be retrogradely transported to neurons in the prelimbic cortex, cause the proper expression for downregulation of GR within neurons that project specifically from the prelimbic cortex to the nucleus accumbens. And Princess has shown that she can successfully do this. These are complicated surgeries. She can do it. She can get knockdown of GR here, which is shown here in orange in the transgenic, the flox animals. And when we look at, we essentially knock down GR specifically in this pathway, and then we look at the acquisition of sign and goal tracking behavior across 5 sessions. And when we do this, what you can see is that in orange here, the experimental animals, the flox animals, where we're knocking down GR, they have an increased tendency to sign track. In green here are the wild type animals, where you can see quite a distribution between the tendency to sign and goal track. So again, by knocking down GR in the PRL to nucleus accumbens pathway, we seem to be increasing that tendency to sign track. So what these studies show us then is that GR within the corticostriatal pathway, that from the prelimbic cortex to the nucleus accumbens, may essentially be acting as a brake on incentive motivational processes. The next step then is to determine the mechanism by which this occurs. And our thought is that this is likely dependent on the effects of GR knockdown on dopamine in the nucleus accumbens, and that will be the next step that Princess pursues. So again, back to Pavlov, I've told you that dopamine seems to be involved in the excitatory processes. The PRL to PVT pathway may be in the inhibitory processes, as well as the prelimbic to nucleus accumbens core pathway. So Pavlov stated that broadly, he went on to describe and said these two essentially personality traits, and said broadly we can regard these disturbances as due to a conflict between the processes of excitation and inhibition, which he said the cortex finds difficult to resolve. And so I often will cross out the cortex and put the PVT, but the cortex has sort of garnered my interest more so in recent months. And so it remains to be determined whether it's the cortex or the PVT or the nucleus accumbens or some other structure. And I'll just tell you a little bit about what we have going on in this regard to try to figure out what neural circuitry and neuromolecular mechanisms may be mediating the tendency to sign track or the attribution of incentive value to reward cues. So first, Brianna Ramos, who's a neuroscience graduate student as well, in collaboration with Bob Kennedy in chemistry, is using microdialysis in the paraventricular nucleus of the thalamus to characterize the neurochemical output at baseline and then across learning once we establish the phenotypes of sign and goal trackers. And so this is really exciting because we can look at dozens of neurochemicals at once during the process of learning. And the PVT is a very complex heterogeneous structure. And so this will be, nobody has done anything like this before in the PVT, in part because it's technically challenging, but she has it up and running and we're getting some really interesting data. This is just showing you some heat maps here where you can look at rat 105, who happens to be our sign tracker. There's a big difference in activity in general in the PVT, in neurochemical activity in general in the PVT, baseline relative, or sorry, post-Pavlovian learning relative to baseline. So excited about what's going to come from that. Stephanie is a postdoc in the lab, and she's doing some really great studies to help us better define the inputs and outputs to and from the PVT. So essentially she's using not dual, but essentially a quadruple labeling approach, viral approach, so that we can ask, where are these neurons from the prelimbic cortex terminating in the PVT? Are they terminating in close proximity from the neurons from these subcortical regions like the LH to the PVT? And are those neurons terminating in close proximity to those projecting to the nucleus accumbens? So really excited to get a better handle on the definition of the cell types and cellular activity in response to the reward cue in those circuits. And then finally, Eduardo Peru is a brand new postdoc in the lab who's going to undertake the hard task of trying to get calcium imaging set up in the PVT, but also in the prelimbic cortex. So one of the first studies we're going to do, again, due to technical difficulties of trying to get into the PVT, we're going to focus first on the prelimbic cortex and identify cells from the prelimbic cortex that are projecting to the PVT, as well as those that are projecting to the nucleus accumbens, and see how that neuronal activity may change over the course of learning in SINE and goal trackers. So very excited about the additional work to probe the underlying neural mechanisms contributing to individual differences in incentive learning. Oops. So just check the time here. Okay. So now we're going to switch back. I told you that what I think is really especially interesting about this model is not just the ability to parse incentive versus predictive learning processes, but the fact that we can simply characterize these animals based on 5 sessions of simple Pavlovian conditioning and know that they're going to exhibit a number of traits reminiscent of neuropsychiatric disorders. So we wanted to ask whether or not these traits actually map onto anything in human behavior or potentially the development of psychopathology in humans. And so to do this, I teamed up with my colleagues in psychology, Ashley Gearhart and Michelle Joyner, who was a clinical psychology graduate student at the time, as well as my colleagues and collaborators at the Lauria Institute for Brain Research in Oklahoma, Martin Paulus and Jenna Kalazzi, who was a postdoc in Martin's lab at the time. So I was really excited when Ashley and I began talking about these studies because she was all for what I had always wanted to do, which was essentially take exactly what we do in the rats and put it in front of kids and see what happens. Whereas a number of other people who were trying to set these tasks up in humans and especially in adults were using complex computational tasks, which I didn't think were necessarily capturing the simple Pavlovian conditioning or learning processes that I wanted to. So we essentially had these Lego boxes built. These are literally big, giant green Lego boxes where an illuminated lever comes out. We use essentially the same program that we do in the rats. The lever comes out, it retracts, and the kids get a reward in the adjacent reward cup. The study I'm going to tell you about today involved 9 to 11-year-old kids. It was 40 subjects, 24 males and 16 females. So this is using the same, again, I told you we use the same program as we did in rats. So we started out using the same metrics that we do in rats. So this is a composite Pavlovian condition approach index, where if we do the same thing we do in rats, above 0.5 would be a SINE tracker, below negative 0.5 would be a goal tracker. What you can immediately see is that the population is skewed towards SINE trackers. We're essentially getting no pure goal trackers, even though there is variability in the degree to which these individuals are SINE tracking. So essentially we're calling them SINE trackers and non-SINE trackers. And this is just showing you again that we can quantify these behaviors. We can look across sessions and you see a bit of a learning curve, not quite as pronounced as I would like, but a bit of a learning curve in those that learn to SINE track. And the behavior for both SINE trackers and non-SINE trackers is distinct during the cue presentation versus the intertrial intervals. This is to show you what these behaviors actually look like. So this individual is quite inquisitive, checking out how these things work. The lever is going to come out here on top. You can see it comes out and he inhibits himself. He shakes his hand and his head and then goes to the food cup and waits for the reward to be delivered. So he's actually a goal tracker on that trial at least. This individual is quite fidgety. You can see the lever comes out and they are essentially a SINE tracker. So they're interacting, they're pressing the lever, it retracts, and then they go and they get the reward. So pretty remarkable that we're seeing similar topography of the responses in these human children as we do in the rats. What I thought was especially interesting in this study, in this collaboration with Martin Paulus and his group, was that when we looked at the tendency for externalizing behaviors, we saw that sign trackers show greater ADHD-like problems, greater oppositional defiant problems, social problems, greater fear responses, deficits in inhibitory control, and greater negative affect. So very similar to the list of behavioral traits or externalizing behaviors that we have shown in rats. And then we were lucky enough to get some brain imaging done on these subjects during a monetary incentive delay task. So this is another cue reward task that's slightly different than the sign tracking, but essentially tapping into the same brain mechanisms that are modulating cue elicited responses. And so what you can see here in the non-sign trackers is that they're showing differences in parietal activity in response to the cue, where you don't see those differences in sign trackers. But in the sign trackers, you see their differences are in subcortical regions like the amygdala in this case. So the amygdala popped out as being the most different under these conditions, not a region that we've necessarily spent a lot of time on in the rodents, but it goes along with the notion that sign trackers are relying on these subcortical processes to process the meaning of reward cues. So to summarize this section, it seems that we're able to capture individual variability in the tendency to sign track in youth. The propensity to sign track appears to be associated with externalizing behaviors. And in agreement with animal studies, their subcortical circuits appear to drive cue-motivated behaviors in youth with an increased propensity to sign track. And then the image is just showing you the parallels in these behaviors, which again, I think is quite remarkable. So let me just check the time. Okay. I'm going to tell you the take-home messages and then spend a few minutes talking about something I'm really excited about, future direction of my work. And so the take-home message of what I've told you so far is hopefully I've convinced you that the way an individual responds to cues in an environment may be a key determinant of vulnerability to psychopathology. There seems to be this imbalance between top-down cortical control and bottom-up motivational drive that's contributing to maladaptive behavior characteristic of psychiatric symptomatology. And specifically, I told you that dopamine appears to play a critical role in the subcortical motivational drive. The PVT, the paraventricular nucleus of the thalamus, appears to play a critical role in integrating both cortical and subcortical information. And glucocorticoid receptors within the corticostriatal pathway appears to inhibit or put the brakes on incentive motivational processes. And so I fully recognize that we need to be cautious in translation, but I think animal models of behavior are truly critical to advancing our understanding of neuropsychiatric illness. I think we have a ways to go, and I just want to emphasize how important behavior is and how we should appreciate the differences and embrace them, because I think it'll be really telling and informing translational value. So I'm going to spend a few minutes telling you a little bit about a future direction in relation to the use of animal models. And so I'm collaborating with Jacinta Biener in the Department of Psychology to expand this research into a wild primate population in hopes of better understanding the adaptive significance of the propensity to sign track. And so I was very excited when Jacinta and I decided to actually do this. When I initially thought about studying primate behavior in the wild, I thought, I kind of channeled Robert Sapolsky, and I was thinking, oh, I'm gonna, be hiding in the bushes and just looking at their natural behaviors and see what I think is sign tracking. But then Jacinta said we could actually do even better. I am lucky that, whoops. So she and her colleague Tori Bergman, as well as Marcela Benitez at Emory, have this population of over 300 capuchins in Costa Rica, and I'll show you a little bit more about that in a few minutes. But essentially, they've been tracking this population for 7 years. They have daily activity records on them, know everything about their lives, family structures, things like that, social dynamics. But what they also do is they have these platforms placed throughout the forest where the monkeys can just naturally at their will come down and interact with different stimuli. And so they're assessing different types of behaviors. So what we did essentially was build, you can see this box that we had to take in various components through customs. That was a challenge, and then get it set up there, but it's essentially the same rat box that we use. And we have it automated such that when the monkeys place their hand into the food cup, it's going to trigger the program, the lever is going to come out and the program will start, the GoPro cameras will start, and everything will be automated from there. And so why is this important? Well, if you, I think this diagram, which is something we've been thinking about and putting into all of these grants that we're submitting in this regard, I think really highlights the importance of it. So we study laboratory rodents because we have great approaches and techniques now where we can really probe brain behavior relationships more deeply than we ever have before. And that's just gonna keep going in that direction. It's pretty remarkable. We know that we can capture the tendency to sign and goal track in these animals. While I claim that the translational relevance is pretty good, it's obviously limited. These are rats. And what's most important here is that these rats are growing up in an impoverished environment. So we have no idea what the relevance of these sign and goal tracking behaviors are in the real world, or what they look like in a natural environment, or how natural factors may determine the tendency to sign or goal track. So that's one big point of importance. If you look at humans, of course, they have all of these factors in their daily lives contributing to these different behavioral tendencies. They have these very rich social lives that may be impacting what they do in the laboratory on any given day. And while we've shown that we can essentially measure the tendency to sign track in adults, we know we can capture sign and goal tracking behavior. We have relatively limited resolution of what we can look at in the brain and what we can conclude from that. And we also find great difficulty in really tracking all of the factors that may be driving their behavior, again, on any given day in the lab. But of course, the translational relevance is quite high. So then this is why we wanted to look at these wild capuchins. And again, like I said, for over 7 years, they've been tracking these animals. They have their social structures, they know their daily activity, their vocalizations. We can get hormonal data from urine and feces. And it's really amazing the amount of data, the data set they've already amassed is really unique and quite amazing. And so now we can put on these platforms, these apparatuses, to look at the tendency to sign or goal track, and it be able to better determine not just sort of the social determinants and the hormonal and these physiological measures, but the natural determinants and whether or not that changes across development or with the resources available. And so this is just showing you very quickly. I will get through so we have some time for some questions, but essentially this is on the western part of Costa Rica. It's called the Taboga Forest. This is what one of the field site houses looks like. Within this field site house is actually a kitchen converted to a laboratory where we can run samples. This is what the morning drive actually back, we get up and we go out to the field site around 4:30 in the morning, and then this is kind of what the drive back looks like. We go out early because they never interfere with the monkeys, so that's what makes this quite unique from other sites where they study primate behavior. So these are, again, are monkeys living in their natural environment. They come down to the platforms at their will. The humans never interact with them directly, and we never interfere, and they never see us interacting with the platforms or the chambers themselves. And then this is just showing you what the various platforms and the various tasks, they actually, when I was out there, they have touchscreen tasks that the monkeys are performing out there. They have these cooperation tasks, and then this is our box that we are attempting to get set up and going out there. So let's see if this video works. This is just showing you one of the monkeys coming down. We baited it with initially with bananas to get them to come down and interact initially so they can get habituated to it. This is to show you that another monkey will come down, and we'll be able to track all of these things. So we'll know if there's one monkey down, if there's two monkeys, if they're related, how old they are, and you can see them starting to interact with the chamber. Oops. And we're competing against all of these, all of the other normal things they do on a daily basis. So it's quite frustrating for me given the limited time when I'm out there. I've been out there twice now, only for 10 or 12 days at a time. And so each day is really important, and I'll sit there for hours with the monkeys just sleeping above me, doing nothing. And there'll be many days where they don't come down to the chambers or interact at all. But that's what essentially is once we have all of this automated and set up and running, we'll be able to get all of this data and know, what drives them to come and interact in the first place. Is it when resources are plentiful or not plentiful that they have a tendency to sign or goal track? So here you can see a monkey eating the palm fruits during the rich rainy season. And then this is one of our GoPros catching a monkey, and you can see us here in the background watching. And so we have a number of plans, like I said, to look at all of these sort of environmental and natural and social factors and how it might lead to the tendency to sign or goal track. I'm also excited about getting all of the video data in the monkeys, as well as what we already have, I told you, in the laboratory rodents. And we're starting to also implement deep lab cut analyses in humans as well. So with the hope of using a cross-species approach, applying this deep phenotyping to see if we can really identify similarities or differences between species with similar tasks. And then finally, I've already told you that ongoing and future directions in my lab here at Michigan are focused on elucidating the neural mechanisms that contribute to that propensity to sign track. And the human studies here in orange, I hope to eventually conduct longitudinal studies and truly investigate the relationship between sign tracking tendencies and the development of psychopathology. And we're working really hard to secure funding for the monkey work as well. And I'm really hopeful and excited about that. And then with that, I'll leave you with this picture. We have this monkey filling in the blank here in the middle. And I hope next time I talk to you guys, I'll be able to show you what a sign tracking and goal tracking monkey looks like. So I'm gonna, oops, and then finally, sorry, I just want to thank my wonderful team. This was, as I tried to highlight throughout, many people were involved in all of this work. And of course, I couldn't do it without my great team and the collaborators. I'm going to try to stop the screen so I can see you guys. Okay. I'm happy to take some questions.
Oh, that was a lot of fun. Thank you. So, but you haven't studied any pathologic human samples, I take it.
Nope, not yet. And then that's the hope.
Yeah. Okay. Cindy.
Yeah, so Shelly, thanks again for a great talk as always. I want to go back a little bit to the question of exactly what it is maybe that's being inhibited, and also sort of like what people and animals, I guess, are responding to. So, again, going back to like even those first dopamine response graphs that you were showing, it looked to me like even the goal trackers, it's not that they don't have a dopamine response to the lever coming out. It just, it actually looked almost even, and just, like it looked like a kind of like a 40-60 sort of situation. Like they were just spreading that response out over the two events. Whereas for the sign trackers, it really gets concentrated onto the sign itself.
Yeah,
And likewise with the kid too, it's not that he didn't notice it. So is it really the incentive salience that's getting blocked, or is it is it a sort of a redirect, is it an inhibition of the action and response to it and a redirection there?
Yeah, I think the best example to address that question is actually the pharmacological study that we did to follow up, where when we give dopamine, when we block dopamine with dopamine antagonists systemically in that same study essentially that we did the voltammograms with, what we showed was that we are blocking performance of both sign and goal tracking when the dopamine antagonist is on board. But what was really interesting is when we remove that, we show that there's actual evidence of having learned the goal tracking response, having attributed predictive value, but no evidence of having learned the sign tracking response. So that shows you, to my mind, that tells us really clearly that dopamine is involved in incentive learning and not just predictive learning.
Okay. I think Sergey, I think you have a question.
Yes, thank you very much, Dr. Flagel, for the talk. Very fascinating, particularly looking at how you're trying to translate from rats to humans and primates. The moment you showed me the first picture about goal tracking and side tracking, I imagined myself at Domino's and looking at the pizza tracker, and I was like, okay, what most of us are going to do is going to go towards the screen or towards the pizza guy and take our order and get out. So I'll say, or most of us will agree, that 99 or 100% of us are going to go to the pizza guy rather than to the screen on the board. So and then when you showed me the human, the kids doing the task, the outcome was that the kids who were stressed or had a attention deficit or any other sort of mental disorder, they were actually hyper. And even in your coat animal, like when you control the coat and you introduce corticosterone, you see like shift between. Do you think that this all is linked to like stress? like, or it's hyperactivity? Like,
Yeah.
is it like it's a, and one last question on this is like, and it's mostly like, because in humans by default, like if you agree to the top down and bottom up, it's mostly top down for humans, and in a disease state becomes bottom up, something on that line. What are your thoughts on it?
Yeah, great question. So actually, I didn't get into this. I alluded to it a little bit, where I actually think that court in this case is really tapping into reward processes and not stress responsivity. So if we actually look at stress response, whether it's corticosteroid response or behavioral responses in sign and goal trackers in the rats, we don't see any differences. So they don't differ on anxiety-related behavior. They don't differ in their court response to stress. They do differ in their court response to reward, and we can administer court and GR antagonists and affect those reward or cue elicited responses. And so I actually think that the way that there are differences in, stress system, but it's actually those differences that are interacting with the reward processing in sign and goal trackers that are driving their response. In humans, it was actually quite interesting, because one of the factors that came out as a strong correlate of the tendency to sign track was socioeconomic status. And presumably that taps into, early life stress mechanisms as well, or stressful situations. And so that was something, of course, that we can't, we haven't necessarily, well, my lab hasn't studied it directly in rats, although others have shown that you can stress, take rats away from their moms early in life, and increase the tendency to sign track. So there is some interaction there. There's also studies in adults where you can, elicit stress and then see an increase in the tendency to sign track. But I think in terms of the mechanism, and at least the way we're thinking about it, is that GR and court is involved more in that interaction with dopamine and the reward processing than it is a stress responsivity in this model.
Awesome. Thank you very much.
Yeah, thanks. Any other questions?
Any other questions for Shelly? Okay. So that was okay. Well, I think we're just about at the end of the hour or so, but thanks very much. That was a really fun talk.
Yeah,
And
thanks for having me.
yeah, so I think, the monkey study is a really cool idea. I hope you're successful in getting funding for that.
Me too.
That's a really creative thing, I would say. So
Yeah, me too. All right, thank you, everyone. Take care.
okay.